All right, let's go see our data. So go to my data. And you should see it'll be in your, obviously you won't say Michael Edwards private project, but it might be private project. You'll see your gene list here. Click that arrow. Here's what I want you to do. Let's add that now to our meta-analysis. Now we can see how cool our gene list is compared to 6 other ones. Here's what happens is people run their experiment and they think that like this is the only way to like view things. If 6 other people did the same thing you did, you should listen to them as well, right? That the best of all those studies is an average of all of them. So click on the plus. Now click on this window here. It says quick view. Click on this for our gene list, right by your gene list. Click on that. So now what we're doing is we're using our gene list as kind of the seed for basically this program. The first window will be the quick view. It'll give you all the genes on our gene list. What's the #1 ranked gene in this gene list? CD44. Anybody heard of CD44 before? Yeah. When you ranked him, remember the SeqMonk ranked it by p-values. Does anybody remember what the top gene when that was at? It's like BCL2 like. 281. You always correct me. Yeah. Why is it ranked differently? I think this is one thing I really like about the BaseSpace, the correlation engine, is that it ranks them based on your fold change and p-value, right? So it'll take the absolute fold change and it'll rank those, and then it'll rank your p-values, and then it kind of takes an average of both of those and uses that number to rank these. Everybody see that? You can tell, look at CD4's fold change in our activated state. That's huge. Basically what this is telling me is that in the germinal B cells, there's probably nothing of this, and then once they become activated, there's a ton of it. I now have a marker for activated B-cell lymphoma. Not only do I have a marker, I have potentially a way to pull these cells out of any kind of mixture. CD44 is on the outside of the cell. Maybe this is a way I can target those cells. Everybody see that? Look at the next one. If you mouse over it, it'll tell you exactly what gene that is. What is that? Glutamine. I now found its food source. I know how it eats, right? And that's just me ranking the genes a little different. We're coming back to CD44. Hold on. But I want to show you some other things. We can go to curated studies. So this will tell me what studies in their database most look like my gene list. That is powerful. And that is something IPA does not do. So I've got a bunch of my gene lists up here. Of course, they look like that. But go to your first one. 2 major subtypes of diffuse large B-cell lymphoma. And click that blue arrow. You'll notice sometimes as you click windows, you'll open another window. If you get lost, look on one of your other tabs, and you'll find your original window. So it's going to kick you out here. I want you to click on full study details. Look at the paper. What paper is that? Science! Dang straight. Here's the deal is, go back to, so this is a new, go to your other window. Click on this red arrow here, or I think you can click on this. Just click on the diffuse, the title. This will tell you. So here's our gene list, and here's their gene list. It's the same study. But again, we had 161 that were common. But here's where they're telling you is if it went up in our study, all the genes that went up in our study went up in theirs. All the genes that went down also went up in theirs, and nothing was contrary. That's why the p-values, if you look at the p-value, the probability of getting this by random chance is 9 times 10 to the negative 89th. Those are the same odds as David Bowie and Prince giving a concert next year. Is it too soon? Oh, come on. And I'm fans of both, by the way. I'm a huge Bowie fan. I love Ziggy Stardust. So, all right.
But if our gene list was extracted out of that paper, why is there this difference?
What did we do in our study? What did you guys do? When you were setting up your t-test, what happened?
Oh, 0.05.
No, you kicked one out, right? They used that crappy sample in their study. That's why our gene list is better. We're getting more genes because we kicked out that crappy study. And they included it and they published it in the paper. No one would ever know unless you actually looked at the data. Like me and, basically people in this room are the only people that know that probably. That happens to me all the time. I go through these studies and there is absolute garbage out there. And people publish in really big journals. Kills me. Okay, so again, this will give you an idea. You can go down here and obviously it's going to pull up a lot of things that are associated with cancer, and we would expect that, right? Even mutation. Well, actually, don't look at this thing just has all these mutations. But anyway, B-cell lymphoma.
Just one more point. We also did a different type of years. So every time you analyze the data differently, you get a different. So we removed a sample and we did it differently.
And ours is better. We can go to Body Atlas. So say I'm doing this. I'm going to show you one thing here. The body. Oh, I'm running out of time. Go to Body Atlas. Go to click on cell types. So that's that body, the guy going like this. Click on him. It'll cut tissues comes up first. Then you'll see cell types. Here's what I want you to do is view by ranks. What's the first cell type that comes up? B-cell lymphoma. And it says germinal, and actually our gene list is negatively correlated to it. Our cells are turning away from germinal cells. That's cool. We can also go to pathway enrichment. If I click on this, well, actually, let me go to Knockdown Atlas. Or no, Pharmaco, I'm sorry. So that thing where you see the Pharmaco Atlas, the chemical structure looking thing, click on that. Here's the deal is, okay, I know genes that are associated with the activated B-cell cell type. Maybe now I want to do is find things that maybe would inhibit those genes. Therapy. What do they call it? What is it? Applied or therapy? I don't know. It's a big buzzword now. Everything has to be clinically associated. But so here, this is it. So view by ranks. So click on ranks. So if we're looking for something that inhibits these genes, are we looking for a positive or negative correlation? Negative, totally. Right? Here's what's happening. Those things that are positive, the chemicals that we can add that look like what we're looking at, usually typically, remember STAT3, immunological response. Here's the first thing that's negative. Click on that. Here's where they show that this chemical basically kind of inhibits the genes that we're doing. And sure enough, in the title, it's diffuse large B-cell lymphoma, STAT3. Science. This is not all just, this isn't voodoo. This stuff makes sense. It totally makes sense. We could also, and I'm not going to show you this, but we could go to Knockdown Atlas and we could find genes that affect, if we knock them out, how do they affect the genes that we found changed and activated? Okay, let's go back to our one. So I have a marker for B-cell lymphoma, the activated form. So get rid of this. You can click on this X, get rid of your ABC, CBG thing. Type in CD44 and your gene should come up. Click on that gene. Okay. Again, this is my marker. Now I'm using the Pharmacoa Atlas to find things that maybe reduce that, knock it down. Okay, we can do by mechanism of action, but let's go by view by ranks. Okay, there's a lot of things that upregulate it, and I bet they're all bad. Let's try this second one. This downregulates it. So click on that motoxafen, blah, blah, blah. This is giving you, now this took me to the study where it shows that this chemical reduces CD44. And lo and behold, they're working on lymphoma cells. Who would have guessed? This is awesome. Like, you know, the odds of this stuff just coming together like are just astronomical. It isn't if like you're mining all the data. So if you look here, so I clicked on that title, what you'll see is when they add zinc acetate, it's not their miracle chemical that they, you know, their engineered one, this MGD thing. It's only when they add just zinc acetate alone that it actually decreases CD44. Could a potential therapy to activated B-cell lymphoma be something as simple as going to the vitamin store and getting a zinc supplement? I was looking, I actually found some papers. It is known that zinc levels are decreased in B-cell lymphoma. Could a supplement maybe affect that? If it could, there's a reason why this probably got dropped because, you know, no one can make money on that. Okay, any questions on BaseSpace? I wanted to show you is that here's what we did with BaseSpace is we searched just by a disease. That first thing we did, we didn't even have our data and we found the gene list. We uploaded our own gene list and explored that, and we can use other gene lists to look at that. Then what we did is we found an interesting gene, and when we use this software to kind of explore what's going on with that gene. This works on all kinds of different levels. Okay, let's go to, I'm running out of time. Let's go to your IPA.